Hello, thank you for having me. Today I will be speaking to you on behalf of the Mexican Biobank project. This project started as a collaboration between Langevio Sinvestad and the National Institute of Public Health in Mexico to consider using the blood samples and link trait and disease data collected by the Institute as part of their year 2000 National Health Survey for DNA genotyping and complex trait analysis. The collaboration extends to Oxford University and University of Leicester in the UK and the University of Chicago in the US, among an even larger collaborative network. I'm deeply honored to have the opportunity as a postdoctoral research fellow to work on the DNA of Mexicans across 32 states and across both rural and urban localities, to learn about the genetic history of this rich, diverse country and to try to shed some light on the impact of this history on complex trait architecture, and to do so respectfully. The project comes at a time when there remains a persistent bias in genome-wide association studies with regard to the genetic ancestry and amidst growing calls to diversify genetic and complex trait databases. Now, why would we want to diversify genetic phenotypic databases, and what do we hope to learn? This question has a difficult history and likely a difficult answer, but we can try to unravel the facets here. Genetic ancestry has been conceptualized and inferred using methods such as principal components analysis and admixture, to name the most predominant ones, and they capture the way in which we vary genetically from each other due to our near and distant ancestors. So we started by visualizing population structure and calling ancestry using standard approaches, finding known patterns in Mexican history and making some new observations. We visualize the Mexican biobank samples using a principal components analysis and find that the majority of Mexican individuals lie on a cline between present-day Europeans and indigenous Americans, reflecting the history of Mexico following Spanish colonization and subsequent independence. We also observe a pull towards present-day Africans, which reflects the transatlantic slave trade that brought many Africans to Mexico during the colonial period. We compute ancestry in the Mexican biobank using admixture with people from around the world as our reference panel. I have broken out for visualization the Amerindigenous and European ancestries in the two panels below. We see varying degrees of Amerindigenous and European ancestry with higher levels of Amerindigenous ancestry in the center and in the south of the country. We observe that the African slave trade has left its mark across the country, with some African ancestry found in every single state. We observe that only 3 states in the north have more European ancestry than Amerindigenous ancestry, with Amerindigenous ancestry being the majority ancestry in every other state. Lastly, we note the presence of a small but significant East Asian ancestry in almost every state, the highest in Guerrero, and even more modest but significant amount of South Asian ancestry in the majority of states as well. We note that this East Asian ancestry has been previously observed genetically by Juan Esteban as a master's student in the Moreno lab, and has been documented by several recent historians as they uncover the forgotten history of Mexico's Asian population. The story goes that these Asians arrived from the Manila port in the Spanish Philippines to the Acapulco port in Mexico through the 16th and 17th centuries, with origins as diverse as the Philippines, Indonesia, Malaysia, India, Bengal, and Sri Lanka, among others. However, they were collectively referred to as Chinos by the colonialists. There have also been later 19th and 20th century migrations from China and Japan, especially to the north of Mexico, which explain part of the Asian ancestry we observe in the biobank today. While I will not have a chance to go into this in detail today, we used an ancestry-specific PCA approach developed in the Moreno Estrada et al. To genetically pinpoint the origins of the East Asian, South Asian, and African ancestries in the biobank, and find results corresponding to the historical record as illustrated in this example for individuals from Guerrero with high South Asian ancestry. Some of these ancestries reflect a sex bias, especially the predominant Amerindigenous and European ancestries. To learn more about that and networks of Y chromosome and mitochondrial lineages in Mexico, please see Maria Jose Palma's poster. Now, these ancestries are differentially distributed in different individuals and in different states. And we observe the largest genetic differentiation as measured using FST along a north to southeast cline. Each of these ancestries reflects a specific demographic history. To understand the demographic history of contemporary Mexico, we first need to see the global perspective. In this map of the peopling of the world as learned through genomics, key to note is that all individuals that left Africa went through a bottleneck or shrinking in population size, followed by re-expansion as they traversed across Europe, Eurasia, and Australia, including present-day Europeans. A second key point is the bottleneck that all ancestors of present-day Americans went through when crossing the Bering Strait. Now, contemporary Mexicans carry predominantly Amerindigenous, European, and African ancestry, each of which went through a specific history of genetic bottleneck. To reveal the more recent history of population sizes of these three ancestries, we use genetic haplotype identity and a method called ancestry-specific IBDNE, which has been shown to work well in admixed populations. And while we can't go as far back as the Bering Strait bottleneck 15 to 23,000 years ago, we can see the dynamics as far back as roughly 9,000 years ago if each generation is 30 years. We use Mesoamerican chronology to color the periods and find, interestingly, that for native ancestry, effective population size went through a slow and steady decline in the classical period, followed by an increase in the post-classical period, and then a smaller decline in the colonial period. We also observed that both African and European ancestries that entered the contemporary Mexican gene pool went through a sharp decline in population size during the colonial period, with the smallest population size observed for African ancestry, and both have since been increasing in population size. Now, this analysis is using identity by descent segments, but we can go one step further and consider runs of homozygosity or ROH. To understand what an ROH is, consider two parents, D and E, shown here, that are identical by descent, as in this cartoon in the second green segment, due to a common ancestor A. The child F will present an ROH by the pairing of these stretches in homozygosis, as shown in green. ROH can be a window into demographic and mating history. Key to note here is that while a bottleneck population, or one that has maintained a small population size, will result in a large number of small ROHs reflecting distant shared common ancestors, consanguinity or marriage between relatives would instead result in fewer but longer ROHs reflecting more recent shared common ancestors. We call ROHs in the Mexican Biobank and observe that both the number of ROHs and the total length of ROHs per individual increases as we move north to southeast in the country. We confirm that this is primarily due to a positive correlation of ROH with Amerindigenous ancestry. So we asked whether Amerindigenous ancestry carries more ROHs due to historical bottlenecks and small population sizes, or due to consanguinity. To answer this question, we look at the histogram of the total length of ROH per individual in each state after first partitioning ROHs by size. We first look at the northern states and the central states, and finally the southern and southeastern states. We see that there are more ROH per individual moving southwards in Mexico as before, but observe that this is in large part due to small ROHs, implying that bottlenecks in small population sizes rather than consanguinity has been largely responsible for more ROHs in Amerindigenous ancestry. Now, ROH have been shown to have associations with a broad range of complex traits, and estimated to have reducing effects on height, weight, and cholesterol, for example. So we consider the role of ancestry as may manifest through runs of homozygosity or other aspects of genetic variation affected by demographic history, and ask whether they're important in understanding variation that we observe in complex traits or disease. For example, why some people are taller or more likely to have cholesterol. We also realize that not only does genetic ancestry mean the experience of a different demographic history, but also that of a different environmental history. Further, the environmental present, as is manifested in experience of discrimination, socioeconomic opportunities, and living in a rural or urban environment, can vary independently across individuals, but is often correlated with genetic ancestry due to the particular historical contingency of a European colonization, of which many remnants still remain in a post-colonial Mexico. Keeping all this in mind, we first look at the distribution of average height across Mexico and find that it increases roughly south to north. We're interested in modeling the distribution of this trait using a set of potential predictors. These are either computed from genetic data such as, or from linked demographic and life history data, and include genetic ancestry, ROH, as well as socioeconomic status, living environment, and altitude, among others. We further model true random effects to capture the covariance structure defined by the relatedness in the data, and to capture any unmodeled environmental variables that may vary from state to state. We'd like to understand which of these predictors are important for variation in height, and would like to estimate an effect size for each of these predictors on height. We run this analysis as a linear mixed model, and we observe the following: that amerindigenous ancestry has a height reducing effect, while higher education attainment, living in an urban environment, and African ancestry have a height increasing effect. Interestingly, even though amerindigenous ancestry has a height reducing effect, if we look at height as a function of birth year, after first dividing individuals into 4 groups by their level of amerindigenous ancestry, we do see that individuals with higher amerindigenous ancestry have lower height, but that all groups are increasing in height due to the effect of changing environmental factors in the last century. I've also modeled a range of other traits similarly, but because of time constraints today, would only be able to say that each trait has its own profile for whether genetic ancestry affects its variation and in which direction. Lastly, we looked at the impact of demographic history on genetic variation at the ABO locus, which has been associated with having severe COVID symptoms or respiratory failure. We find that amerindigenous ancestry is protective against severe COVID, as may be caused by the ABO genetic variant. This can be seen as the frequency of the ABO risk variant decreases with increasing amerindigenous ancestry. In conclusion, we find varying sources of ancestry in Mexico in light of its unique history, infer population size changes and runs of homozygosity using genetic haplotype identity, and observe correlation between ancestry and other environmental variables and a range of complex traits such as height and severe COVID risk. This has obviously been an immense team effort, which, while international in composition, is firmly rooted and led in Mexico, and for which I would like to acknowledge the entire Mexican Biobank team. I would especially like to acknowledge my two postdoc mentors, Andres Moreno, for his vision to give birth to and successfully execute such a large scientific project from Irapuato, Guanajuato, and John November for his vital support in thinking about the difficult problem of how diversity affects complex trait variation in historical context and helping translate that into methods and analysis. I'd like to end by giving a shout out to my research group starting in February 2021 at the Centro de Ciencias Genómicas of the UNAM, where I'll continue to work on questions like these. My lab will be a place where we will not only learn from the scholarship of geneticists and biologists, but also from that of historians and social scientists, whether it be to complement or inspire in investigations into genetic history, or to help us learn about the history of our own field and the concepts that we use today, so that we may do conscious and conscientious science for a more just world. If this sounds like it may be of interest to you, please reach out to me informally to discuss research opportunities at different levels. And with that, I'm happy to take any questions. Thank you for listening.